Chapter 2. Cells, tissues, organs, and organ systems. What are cells? Okay, two different kinds, primary kinds of cells. You're looking at prokaryotic and eukaryotic. Prokaryotic cells have no nucleus, and they also lack membrane-bound organelles. Those are going to be found in two of the domains, eubacteria and archaea. Other kinds of cells are eukaryotic cells. These are going to be larger. They're going to have membrane-bound organelles and nuclei that are going to contain DNA. Everything is going to happen at a cellular level. It's going to be magnified from there often, but it is going to happen at a cellular level. From the cellular level, living organisms, it's going to go up. So cells to tissues to organ systems to animals. But we're going to start down here at cells. Cells are the basic functional unit of life, as I've already mentioned, prokaryotic and eukaryotic. Eukaryotic cells have three basic parts. Plasma membranes, those are the outer boundaries of the cell. They separate inner metabolic events of the cell from the environment that is around them. Cytoplasm is part of the cell outside of the nucleus. It contains the semi-fluid cytosol, and within the cytosol, you're going to find the organelles. Then we have the nucleus, which is basically the control center of the cell. It contains chromosomes. It's separated from the cytoplasm by the nuclear envelope. This is a more detailed comparison of prokaryotic and eukaryotic cells. Here you will see a very traditional depiction of a generalized animal cell. You will find that there are differences in some of the structures between animal and plant cells, which are why you have to be specific that this is an animal cell. Why are most cells small? Well, because the volume of the cell's nucleus relative to the volume of the cytoplasm must not be so small that the nucleus can't control the cytoplasm. There has to be some level of communication between them so that it's restricted. The cell size is restricted. As the radius of the sphere, the cell that is, increases, the volume increases more rapidly than the surface area does of the cell itself. So if you look here, so as the volume increases of the cell, the surface area doesn't increase as rapidly. So if the volume becomes too large, then the proper exchange of waste and nutrients can't happen, and therefore the cell size is restricted. You notice again, just go back into something that I touched on with the first lecture. I'm just zipping past the learning outcomes. They're still there so that when you can go back and review the lectures, PowerPoints by yourselves. They're still there and you can refer back to them in the chapters, but I'm not going to discuss them in detail. Okay, cell membranes composed mostly of phospholipids. Lipids, as some of you may be aware, are fats and proteins. Those proteins allow certain materials to move across them and not others. That means that they are selectively permeable. So permeable being that things can get in and out, selectively meaning that not everything can get in and out. Cell membranes regulate the movement of material, so they are in control of what comes in and what goes out of that cell within some reason. Of course, some things like pathogens, viruses, bacteria may be able to get in and out. They do separate the inside of the cell from the outside of the cell. They separate also an organelle sometimes within this from the rest of the cell. So organelles are like miniature organs. They also provide a surface area. So in certain circumstances, on the surface of a cell, there's going to be receptors for things like hormones so that reactions can take place when necessary. Those sites for receptors right here. And they're going to separate cells from each other. So if you look at a diagram of a cell membrane, this is what you're going to see. So here you will see two layers of phospholipids. They are bipolar. And they're charged, excuse me. They are charged. So you'll see them tails together. What you see, these big things are globular proteins. Those are transfer proteins, transport proteins. So certain substances can get in and out of the cell membrane through those transport proteins. Larger molecules can get through there. Movement across membranes. There are multiple ways in which things can go across membranes. Several things don't take energy. If you're moving from an area of high concentration to an area of low concentration, then no energy is going to be needed. So that's like if you dropped if you took a sugar cube and dropped it in coffee, it's naturally going to diffuse throughout the coffee. Generally, you don't want to wait for the coffee to drink it, so you're going to stir it to make it go faster. But if you let it sit there, it should naturally diffuse from that high concentration area throughout the coffee to the areas of low concentration. That's simple diffusion. And that is, and there you hear my dog with his collar. That is what's depicted here. So cube in the bottom, it's going to naturally diffuse out. Osmosis is simple diffusion of water. It's just a special circumstance of diffusion. That's all that it is. Facilitated diffusion is when a particle's involved. For filtration, you're looking at particles going across a membrane, things like blood pressure. For blood pressure, because of that force of the fluid forcing particles through a membrane. Active transport is a different situation. With active transport, you're going against a concentration area. So you're actually going from lower concentration to higher concentration, which is not a natural order of events, and therefore energy is required. Energy within a cell comes in the form of ATP. Adenosine triphosphate is what that stands for, which is why we just call it ATP. So this involves carrier proteins, those big globular proteins that I pointed out in the cell membrane. Here they are. So those are going to transport whatever is moving from areas of low concentration to areas of high concentration, and that does take energy. Additional forms of movement are things like pinocytosis, phagocytosis, receptor-mediated endocytosis. In general, endocytosis, exocytosis, all that means is it going in or is it going out? Endo's in, exo's out. These pinocytosis, phagocytosis, what those are is groups of particles. Not just one particle, so a group of particles moving in or out of a cell. Okay, the rest of the cell. Cytoplasm has two parts. I already mentioned the cytosol, which is the more liquid part of it. There's also the cytomembrane system, which I thought I had a picture of, but I don't see it here. That's going to be the rest. Ribosomes are where protein synthesis takes place when it's on the endoplasmic reticulum. So endoplasmic reticulum comes in two forms. You can have rough ER and you can have smooth ER. Rough ER has ribosomes on it. So it looks like this. Smooth ER looks like this, has no ribosomes on it. So these ribosomes are making proteins. Okay? Golgi apparatus. Golgi apparatus looks like more or less like a stack of plates. And what it does is it packages, processes, and transports. So it will take anything that needs to be transported out of the cell, package it appropriately, and shoot it out of the cell. It will also take digestive enzymes and shoot them at any point in the cell wall where something is trying to penetrate it as a defense mechanism. So it's like a little militaristic gun. It will point itself appropriately at the threat and start shooting digestive lysosomes at it. There's a Golgi apparatus. So if you can imagine that this little word right here is a threat, the Golgi apparatus starts shooting lysosomes at it. Lysosomes, containing digestive enzymes, digest nutrients, clean away the dead and damaged cell parts, and they also do act somewhat in defense. Okay, mitochondria. These are the powerhouses of the cell. They take ATP and convert it to energy in the form that a cell can use. Your body can adapt to your condition. So in other words, if you are increasing your energy needs, increasing your endurance, your body is going to respond by creating more mitochondria. This is mitochondria. Microtubules and the filaments. Here's the picture I was referring to. So here we have the microfilaments, actually supporting within the cell some of the organelles. So right here it has a little mitochondria. Cilia and flagella, not on all cells, on some cells, on the surface, and they usually function in movement. Occasionally can act as signal. Flagella tend to be longer and whip-like. Cilia tend to be more like little hairs coming off. Okay. Nucleus has a very large job. Contains the DNA, controls all activities, both metabolic and genetic. The nuclear envelope is connected to the nucleus, and it allows things to go in and out of the nucleus. DNA, however, is too large to fit in and out of the nucleus, so it has to code for genetic information on RNA. The DNA does code for regulating cell activities. Okay, now up from cells, level of organization. We go from cells to tissues, to organs, and then to systems. So you would have like epithelial cells to tissues, to an organ such as a lung or a heart, to a respiratory or circulatory system. There are different types of tissues. Epithelial tissues are going to be surface of the skin. Connective tissues, your blood is actually a form of connective tissue. Muscle tissues, nervous tissues. I'm not going to spend a lot of time going on over different kinds of tissues. I recommend you read through this, the different kinds of tissues and what they do. I will never ask you to identify the slides, but they're here. Just read through them. And then organs. Organ is a functional unit made up of more than one kind of tissue. So I'm guessing many of you can already identify multiple kinds of organs. Your skin is actually your largest organ. They give multiple examples. Organ systems are highest levels of organization. So an organ system is something that serves a similar function. So they're doing a certain purpose. Respiratory system serves a purpose and a set function. Here's your list of organ systems. All right, so therein lies chapter 2.